Okay, good evening Gautam, good evening Saima, good evening. Okay, good evening Savita. Okay, good evening Akshay, good evening Shreya, good evening Anirudh, good evening all. Okay, and let's continue the quiz. So what I am saying is, so once the question gets over, please let me know by saying yes or no. By that I can move to the next question. Okay. For every single question, depending on the length of the question, I will give 45 seconds to the 1 minute. Okay, and once again I'm reminding to all of you, I am conducting the quiz mainly, you will become habituated to the time in the examination hall. So always remember, good evening all, good evening. Always remember friends, for part B question in our CSIR NET exam, at least try to spend 30 seconds of quality time. And for the part C question, at least try to spend 1 minute of time minimum. By that you can understand the question, analyze the question in a proper way. Okay, good evening Sujai, good evening Kavita. Okay, let's start. Let's go with the question number 1. So please type yes or no. Okay. Okay, just type let's start. Just type start, we will go it. Just type start, we will go it. Just type start, we will go. Good evening Aditi, good evening. Just type start, we will go. Perfect. Tarun, I want to tell it's hearty congratulations. You are the winner yesterday. Okay, I am requesting all of you, especially Tarun, please join our Telegram group and please send your details to our email ID. Now you will get the details in the chat box. So please send your details without fail. Okay, you are the yesterday quiz winner. Okay. Okay, let's start the question number 1. Okay, here go. 1 minute. Let's start. 1 minute. Let's go it. Okay, I'll come out of this first of all. Yes, go ahead. 40 seconds more. 40 seconds more, friends. Think and answer. Think and answer. Don't be hurry. Don't be hurry. 20 seconds more. Don't be hurry. Don't be hurry. We should never use the word sister chromatids. 10 seconds more. Stop, 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 stop. I think Pechimutu is the only one who has given the answer correctly. Okay, the correct answer will be C, which is a replicated chromosome, as well as friends, which we can call it as a bivalent. So what we will get is a replicated chromosome contains 2 chromatids, while a bivalent contains 4. So we will always say a bivalent. Please kindly do not forget this particular statements. No problem, no problem. Let's move to the second question. I will tell the time. Here you go. Start the question. 60 seconds. Question number 2. They are asking which of the following is not correctly matched. They are asking not correctly matched, friends. 40 seconds. Thirty seconds more. 20 seconds more. Oh, great, 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 great. 10 seconds more. 5 seconds more. Please stop. Good. The answer will be C. It's a very direct question. You don't need to use your brain also, because acrocentric they have given that the chromosome arms are identical in size. No, not at all. They are not at all identical in size. They are always located near one end of the chromosome. So which is not correctly matched is, friends, C. Look into the Shreya, slowly and carefully give the answers. Okay, here goes with question number 3. Your time starts now. 1 minute with the question number 3. Very important one, friends. 1 minute. Yes, yes, yes. 30 seconds more. 30 seconds more. 20 seconds more. 20 seconds more. 20 seconds more. 10 seconds more. 5 seconds more. Stop, 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 stop. Type your answers. No, no, friends, it's a very easy question, right? We read many a time. Anyway, first the answer will be option A. You know very well, we read many a times. What is RB protein? Protein retinoblastoma, which is one of the first identified or first discovered tumor suppressor gene. We read many a times that the RB is basically acting on what, friends? It is exclusively acting on the transcription factor E2F. You all know very well what's the function of E2F. E2F is the major transcription factor which are involved in the transition from G1 phase to the S phase. The E2F transcription factor involved in the synthesis of proteins which are required for the cell to enter into the S phase. So now, suppose example, when the cell get damaged, we have to stop this particular transition. Then immediately what's going to happen? The protein retinoblastoma PRB will block this transcription factor. When it blocks the transcription factor, there is no way the cell is not going to enter into the S phase. The cell never undergo the division also. Hence the correct one is what? A. Good evening, Sumaya. Please continue. Okay, friends, question number 4. This is a very logical question. It's not logical question. You might did not read this. Okay, your time starts now. Question number 4. 1 minute. Continue, Sumaya. Question number 4. 1 minute. "40 seconds more. Think and answer, think and answer, think and answer. "20 seconds more. We read it actually. You require little intelligence. 15 seconds more. 5 seconds more. Stop. So friends, what's the question asked? Anyway, first the answer will be A. The answer is A. What is the question asked, friends? 19S regulatory particle facilitates. That's the question asked. Every one of you has to remember, we read in the cell biology chapter many times, the 19S, it is actually a part of 26S proteasomal complex. We read many a times. What is the function of 26S proteasomal complex that we read in our classes, friends? I told you many a time, 26S proteasomal complex will start degrading the protein which are ubiquitinated. So it is degradation of polyubiquitinated proteins. So whichever the proteins that are associated with the ubiquitin molecule, that can be degraded by the 26S. So this 19S is the part of 26S. Very good. Perfect. Perfect. So get ready for the question number 5. Get ready for the question number 5. Friends, you all can go with the explanations. If you have any kind of doubts, you can ask me, please. Yes, question number 5. Your time starts now. 1 minute. 1 minute. Forty seconds more. Oh my god, so fast, Gautam. Okay. 30 seconds more. Saima, Kavita, Sumaya, good, Akash. Oh, that's great. 20 seconds more. Today is so nice. Everyone is giving very fast. That's great. 10 seconds more. Great, great, great. Stop. Type your answers. Okay. Anyway, first let me tell the answer. The answer is A. So you all know, right? Frapp technique basically explains us the arrangement of the proteins. You know, right, friends? Proteins can move basically what? Laterally. So the lateral movement of protein within the membrane can be easily understood by using this particular Frapp technique. Very, very good. It's a direct question. The answer will be A. Okay, 1 second, 1 second. Get ready for the question number 6. Question number 1. Sure, Shreya, I will explain the question number 1 at last. Don't worry. Okay, friends, question number 6. Your time starts now. 1 minute. Question number 6. 40 seconds more. Twenty seconds more. Fifteen seconds more. 10 seconds more. 5 seconds. Stop. Okay. Now the answer for the question number 6 will be the B, which we call phosphorylation. So we discussed, look into the given question, the biochemical reaction that is most commonly utilized by the living cell. What's the question asked? To propagate intracellular signaling. You know, right, friends? Intracellular signaling means what? Most of the enzyme linked receptor you know very well. Best example, let's take the RTK. How they can show their response? By the process of phosphorylation. Why? Because their cytosolic domains always undergo the process of autophosphorylation. By the process of autophosphorylation only they can show the signal. Okay, that's good. Please be ready for the question number 7. Yes, don't be hurry, don't be hurry. Your time starts now. 1 minute 30 seconds. Correct, Shreya. 1 minute 30 seconds. Your time starts now. It's a big question. Question number 7, 30 seconds. Option P, diacylglycerol. Option Q, Krebs. Option R, GTP. Protein kinase A, RAS protein, phospholipase D, and protein kinase G. One minute more. 1 minute more. Oh, so much fast. Okay. Look into the question, please. Look into the question, please. Don't be hurry. Don't be hurry. 40 seconds more. Slow, 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 Shreya. Slow, Akash. Analyze the question correctly. 30 seconds more. Tarun, Savita, where you are keeping your brain, huh? Think it correctly. Slow, Savita, why you are taking tension? 10 seconds more. 5 seconds more. Stop. Friends, the correct answer will be C. It's a very direct, a very straightforward question. Correct, na? DAG, diacylglycerol synthesis. You all know very well how the diacylglycerol can be synthesized. We discussed many a times the phospholipase C, which is an effector molecule. It will break down PIP2 into IP3 molecule into DAG molecule. Then what we will get? Then what is happening? We will get the DAG. So what's the question asked here? Diacylglycerol synthesis that can be mediated by phospholipase. I have told you phospholipase C alpha, gamma, beta, beta 2, whatever it may be, but it may be phospholipase. Next one, Krebs phosphorylation. We discussed Krebs. What is Krebs? It's a transcription factor, cyclic AMP response element binding protein. Who's gonna activate this particular Krebs? Cyclic AMP activate. Cyclic AMP activate the protein kinase A. Now the protein kinase A activate this particular Krebs. Then the Krebs basically show the response. So the Krebs phosphorylation is mediated by the protein kinase A. Very simple direct question. So Q will be the 1, P will be the 3, and GTP hydrolysis. What you have to understand? RAS protein. RAS, whenever it is active, it is in the form of GTP. RAS, whenever it is in the inactive form, it is the GTP. So the correct answer will be C. Slowly, friends, slowly think and answer. R can be 5. No. How R can be 5, dear Shreya? That's not at all possible, because you have to understand the concept of what we can say cyclic AMP signaling pathway. First, cyclic AMP activate the pKa. pKa enter. Please kindly look into this once again. I have given the explanation also. pKa will enter into the nucleus. It will add the phosphate group to the Krebs molecule. Then the Krebs molecule will come and activate a gene called as CRE. It will come and attaches to this particular region, cyclic AMP response element. Then it will synthesize a lot of enzymes which are involved in the production of breaking down of glycogen into glucose. And okay, as you said that the protein kinase G, I hope all of you remember we discussed in the class. Protein kinase G can be activated by what? It can be activated by cyclic GM, not the cyclic AMP. Cyclic GMP is the one which basically activates this. Okay, perfect. No, dear, no. GTP to cyclic GMP, correct. But where we discussed the cyclic GMP here, the Krebs can be activated by cyclic AMP. Okay, Krebs is not activated by cyclic GMP, Shreya. Hence the correct answer will be C. Okay, get ready for the question number 8. I have given the explanation also, Shreya, you can read. Your time starts now, 1.5 minutes, 90 seconds. Think and answer. Think and answer. It's a logical question, friends. They are asking which is correct. You can ask your doubt, Shreya. But the answer is C. There's a no doubt. You can read the explanation also. Still, if you have doubt, you can ask me. Friends, 1 minute more. It's a little logical question. You have to think and answer. I have given a good explanation also in the next. No problem, guess it. 30 seconds. Oh, someone has given fast answer. Gautam. Okay. 20 seconds more. Then Akash has given 10 seconds more. Saima, Tarun. Good evening, Deepika. Please continue, dear. 5 seconds more. Stop. Okay. The question says membrane proteins are synthesized on the ER also and transported to various cell organelles. We read many times whatever the proteins that are synthesized in the endoplasmic reticulum, majorly where will go? It will go to plasma membrane also. It will go most importantly to the lysosome also. That is the first statement. One hypothesis for the, anyway, first answer will be A. One hypothesis for membrane protein sorting is hydrophobicity matching. That is, protein with a shorter transmembrane portion into the thinner membranes. You are given the following 3 observations. What was the observation? It was found that transmembrane portions of protein in Golgi membrane are shorter than those in the plasma membrane. Presence of cholesterol increase the thickness of the bilayer. And option C, the phospholipid composition of Golgi and plasma membranes are same. And they have given the 3 statement and they asked us which is correct. Friends, you have to always remember whatever the proteins that are present on the plasma membrane, they are having longer transmembrane portion. Why? Because always keep it in your mind, every one of you, whatever the protein that is embedded in the plasma membrane, their transmembrane domains are very larger. Let's tell you one good example of GPCR. Can transpan the plasma membrane how many times? 7 times. The best example you can take. Even certain proteins can transpan the plasma membrane 10 times. You take an example, glycoporine. At least it can transpan 1 time. But friends, whatever the proteins that are present on the Golgi membrane, they cannot transpan especially. Remember once again, the proteins which are present in the Golgi membrane, they cannot transpan the membrane. Very good. So, hence the correct answer will be A. There is no doubt about it. They said that the proteins present in the Golgi membrane are long transmembrane. No. Proteins of both Golgi and plasma, no. First of all, cholesterol is not responsible for any transmembrane ability or thickness. What is the function of cholesterol? Cholesterol is exclusively responsible for fluidity. That's all. Hence, the correct answer will be A. It's a very simple question, little logical. That's all. Okay, friends, ready for the question number 9. Your time starts now. 1 minute. Question number 9. Correct, Shreya. 3 to 12 times banned. Correct. Aquaporine also. Good. Good. Serpentine. GPCR. 45 seconds more. Simple question it is. 30 seconds more. Hope everyone give me the answer. Oh, so fast. Sumaya, look into this, look into this, look into this. 9 is B, 9 is B, 9 is B, 9 is B. Stop. Okay, good. Answer is B. Basic amino acid residues and are located at the C-terminal domain of the protein. My friends, I am just telling to all of you, keep it in your mind. Suppose example, if they have given the same question and if they asked you in the form of a combination, Saima, your statement is also absolutely true, dear. There is no doubt about it. Okay, basic amino acid residues and may be located anywhere in the protein sequence. Will help. It doesn't matter that it has to be located only at the N-terminal domain. It has to be located at the C-terminal domain. Okay, so it can locate anywhere in the polypeptide also. Hence, for this question, I am giving the answer. The people, whether you choose B also correct, you choose also D also correct. For this particular question, I am making the answer as B. But if they ask you combination, you can choose D also. There is no doubt about it. Right. Answer will be B. But D is also right. But B is more appropriate actually, because the reason most protein contain the nuclear localization signal C-terminal domain, but not all. It can be present anywhere also. That's why D also correct. The most appropriate will be B. Please be ready for the question number 10. Question number 10. Your time starts now. 1 minute. We discussed this in the classroom also. "40 seconds more. "30 seconds more. "20 seconds more. Okay, it's a simple question. Think and answer. 5 seconds more. Why you people are taking lot of time? Stop it. Savita. Why? What happened? Saima, Anirudh, Akshay, Sumaya. It's such a simple question. Where you're keeping your brain? Answer is A. I have told more than 15 times in the classroom. Why you people are not answering the question? We discussed, right? Cytosol using outer and inner membrane translocases Tom and Tim, and in the inner matrix, especially TIMS. You know, right? TIM 22 is present, TIM 23 is also present. Correct, na? TIM 23 will be responsible into the matrix. TIM 22, it is responsible for the inner membrane, and Tom will be exclusively located where? The translocase on the outer membrane. Everyone listen. Tom is on the outer membrane, TIM 22 in the inner membrane, TIM 23 in the matrix. Please don't forget these questions. They are very simple. You have to remember how many times I have discussed this. Don't forget. Friends, whichever the questions we can do it, you should not make it wrong. It's not the matter how difficult the question it may be. It's the only matter whether we studied the concept or not. If you study the concept in the right way, how difficult it may be, with a simple analysis you can answer the question. Be slow, thoroughly answer the question. Don't be hurry. The answer for the question number 10 is A. Don't be hurry. Please be ready for the question number 11. Your time starts now. 30 seconds. 30 seconds. Kavita, I will come back, dear. Kavita, I have given the explanation also. I'm sorry I did not see your message. You look into the explanations, Kavita, once after the class you can ask me. Please look into the explanation. 10 seconds more. Anyone for this? Question number 11, D. Oh my God. No one. Akshay, Tarun, Gautam. Yes, other Sumaya. Arey yaar. Saima, what happened here? Anirudh, Savita, Kavita. How many number of times we read? Rotinol is a electron transport blocker, right? In PPTs, in the classroom specifically we discussed, right? Where you are keeping your brain? Oligomycin is what? Oligomycin is an antibiotic which is acting on the F0 F1 complex, which means that oligomycin is, see, listen, be careful, peacefully give the answer. ATP synthase enzyme. It is acting on ATP synthase enzyme. Rotinol, rotinone, antimycin. See, I think you got confused between the rotinone and rotinol. Both are involved in blocking the electron transport chain by acting on the different electron acceptor molecules. Even antimycin also, oligomycin is the only one which is not acting on the electron acceptor. You know very well, F0 F1 complex or ATP synthase enzyme is not involved in accepting of any electron. It is required for what? Through this protons can move, and it is involved in the synthesis of ATP molecule. Correct na? So what is the point you have to remember? This is the only one which cannot block the electron acceptors. It is only blocking the ATP synthase enzyme, that is F0 F1. No problem. Now you remember it. Slowly friends, slowly. Peace of mind is very important. Slow. The more cool we are, you can answer the question easily. Okay. Please be ready for the question number 12. I have given the explanations. You can see here. If you are not satisfied with my explanation, you can easily answer. You can easily understand. Question number 12. Your time starts now. 1 minute. You have to see only 2. By that you can answer. Ten seconds more. Fantastic. Very good. Very good. What is that answer? SKL signal. Very good, friends. Very good. So what are those? Serine, lysine, then followed by the leucine. These are all the 3 signals which are, what do we call it? This as a peroxisome targeting signal. We shortly call it as PTS. Sometimes they'll give PTS. Okay. So obviously it will be SKL. Very good. Very good. The answer will be C. Fantastic. Slowly analyze the question. Okay, please be ready for the question number 13. Your time starts now. Question number 13. Here you go. Slowly, friends, slowly think and answer. Correct, Shreya. Correct, dear. C terminal. If you recall our lesson, you could answer this question dead easily. Anirudh, you have to give the very correct answer for this question. 30 seconds more. I wish you people will give me the correct answer. Mega, check your answer, dear. Check your answer. 13B. Okay, D. 10 seconds more. 5 seconds more. Stop. I think who has given the first answer? I feel Mega. That's good, dear. The answer will be B. We read very clearly now, this is the membrane of mitochondria, right? So here, especially here, so what is happening, friends? Whatever the protein is located here, to that protein, so all the sugars will come and attaches here, right? So where all the proteins are, sorry, sugars are adding, they will add to the lipids even on the proteins also. On the ER and Golgi membranes only inner side. What do you mean by this inner side? Means lumen, which is the inner surface. Very good. The correct answer for the question number 13 is the B. It's a very direct question. We have discussed in the slides also. Okay. Please be ready for the question number 14. Today is really good. Most of the people are giving so nicely. Your time starts now, 1.5 minutes. If anyone can't see the options C and D, I am writing option C and D for you here. Sorry. One second, friends. One second. Just give me 1 moment of time. Okay, for your better understanding, whomever can't see the option C and D, I am writing here. 2, 4 and 5. Option D, 1, 2, 3, 4 and 5. Your time starts now, 90 seconds. Time starts from now. I hope you people will give me the correct answer. It's a very simple question. 1 minute more. 1 minute more. 14 is C. Someone has given. Who has given C? Check the answer. Check the answer. Look into the D also. I have written here. Yes, everyone. Deepika, Tarun, Sumaya. 30 seconds more. Yeah. Stop. Anyone else? Nivetha. Okay. Okay. So friends, look into this. Enzyme targeting to the lysosome involves in which of the following steps? I want all of you has to look into the answer. The answer will be D. Okay. Now everyone answer my question quickly. Savita and Anirudh, answer my question. If a protein is entering into the lysosome, from where it is coming out? From where it is coming out? Savita, where it is coming out? Anirudh, from which organelle it is coming out? It is coming out from the endoplasmic reticulum. Yes or no? So from the endoplasmic reticulum via Golgi complex, the protein is going to enter into the lysosome. There is no doubt about it. So if it wants to come out of the Golgi, first it will, from the ER where they will enter, it has to enter into the Golgi. You are all talking from here. Why don't you talk from here? First, from the ER it has to enter into the Golgi. Whenever it enter into the Golgi, then only it associated with the mannose-6 phosphate, which is acting as a targeting signal. Correct na? Now the point you have to remember, it is getting dissociated from the ER. So ligand dissociation from the receptor of the ER. Don't forget, the protein or anything cannot enter into the lysosome directly from the Golgi. How the Golgi will get? First of all, it will get from the ER, right? ER to cis Golgi, finally it will come to trans Golgi, from the trans Golgi to enter into the lysosome. Now, Once mannose 6-phosphate get added, it can act as a targeting signal. Then immediately what is happening? Once it enter near that particular, what we can say that lysosome, the pH is completely decreased. What happens because of a decrease in the pH? This particular sugar and phosphate can be released out. Correct na? So once the sugar and phosphate get released out, what happens? The particular protein enter into the lysosome. Once again the receptor come out. See here, most of the people has given the answer C, that is option 2, option 4, option 5. I completely agree with all of you who has given the option C. But all of you listen my statements very carefully. Directly it cannot bind to the mannose 6-phosphate receptor. First point, right? It directly cannot attach. First, it has to enter into the Golgi complex. If it wants to enter into the Golgi complex, it has to come out from the ear. So option 1 is right. Option 2 is right. There is no doubt about it. See friends, if something is going to fuse with the endosome, you have to remember, already in case of a endosome, I hope all of you remember, I told you many a times, what is the pH in the endosomes? Obviously acidic pH. Correct na? Where the pH is going to be decreased, it fused with the endosome. Here you have to understand phosphate addition to the mannose in the sense, once it enter into the endosome, it is getting deleted. I mean, it is getting removed. So till it enter inside, it will be added with the phosphate group only. Please kindly do not get confused. That's what, dear, whatever you said is right. But what is the pH inside? They just given the statement before, that's all. Anyway, the pH inside the endosome is the, what we can say that, low only. Now I understand you are saying a statement that early endosome fusion. No. See, why I am using a word late endosome? Late endosome only you can call lysosome. If you use a word, Kavita, early endosome, that cannot be lysosome, dear. You are getting my point? That cannot be a lysosome. Here late endosome means that is the lysosome. You got point? Hence the correct answer will be D. 1, 2, 3, 4, 5. Whomever did not understand, please look into the explanation also once again. Still, if you don't understand, you can ask me in the Telegram group. I will resolve your query. Please get ready for the question number 15. Your time starts now. See, this question you will get it out, sir. Why you have given in the cell biology chapter, you can ask me. Because friends, in our cell biology chapter, we are having a 2E, that is the microbial physiology. In the microbial physiology, we will get this. Okay? Even in the unit 4 also, we will get this. 1 minute more. Here it's a direct question, friends. $30 more. Question number 15. It's easy question. Good, 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 good. 20 seconds more. Perfect. Stop it. Good. So it's a direct question, right, friends? African sleeping sickness. It is caused by what, friends? Trypanosoma brucei. Rocky Mountain spotted fever. It is caused by what, friends? Rubella virus. And mumps can be caused by what? Oh, something got missed. Yeah, mumps caused by rubella virus. Then anything got missed wrongly? No. Everything is correct. Look into this. Answer will be C. The African sleeping sickness disease caused by the Trypanosoma brucei. Whereas the Rocky Mountain spotted fever, it is caused by the rubella virus. And friends, what about this? mumps, it is can be called, something got wrong I have given. One second, something wrong I have given. Oh, so sorry. Answer is correct only. Answer is correct. So P will be what, friends? African sleeping disease can be caused by the trypanosoma brucei. There is no doubt about it. Rocky Mountain spotted fever, it is caused by rickettsia. Correct. Q is the 4. Then the mumps, it is exclusively caused by the rubella virus. Whereas, friends, filariasis can be caused by the Wuchereria bancrofti. Absolutely right. The correct answer will be C. There is no doubt about it. Right? What's the question 4? I did not understand. What's the question 4? Oh, Q should be 4. Correct. Rocky Mountain spotted fever will be caused by the rickettsia. Correct. Obviously, the answer will be C. There is no doubt about it. Is that clear? Understand this question? It's correct. Question number 15 is C. No doubt. Got it, everyone? Question number 15. I'll explain anyone having any doubts. Question number 15 is correct. Okay, get ready for the question number 16. Your time starts now. If anyone having question number 15 doubt, you can ask me, friends. Question number 16. Your time starts now. 1 minute. Choose the correct statement or statements. Please look into this and answer. Please look into this and answer. "30 seconds more." We discussed every single statement. "10 seconds more." Stop. Choose the correct statement. You all know that reverse transcriptase is an enzyme that is used by the retroviruses and retrotransposons. We discussed very clearly. Why, friends? To synthesize their DNA molecule. We know that very well. And so option A is correct. Option B, anyway, answer will be the D. Correct answer. Answer is D. Option B, in human cells, most of the repetitive DNA is present because of the transposons. The same statement also we read while we are discussing the transposon concept. Option C, transposons have no known direct function. Of course, there is a no function of the transposons only. So they just inserted randomly at one particular area and they will only start migrating. So they actually plays a key role in the protein evolution. Hence, friends, option D will be the correct. All of the above. Great. That's all for today's session, friends. Okay, I hope the discussion helped you a little bit. Okay, I feel it helped you a little bit. So tomorrow we are going to start this session at 7:30 p.m. with the molecular biology chapter. Okay, see, friends, actually tomorrow I have to take 2 classes. That is 7:30 p.m. this quiz of molecular biology. Depending on the time availability, at 8:30 p.m. I will start the question paper prediction also. Okay, so that we will see in the tomorrow class. Okay, so please revise once molecular biology for the tomorrow session. Okay, thank you all. I hope it's helped you a little bit. So please revise molecular biology. We will see you in the tomorrow class. Thank you all. Thank you so much. Thank you all. Okay, Shreya, I will provide right now the explanation for you to all the questions. You see it. If you don't understand, you ask me once again. I'll re-explain for you. Okay? Because I have to take immediately another class, dear. So please look into the question number 1 explanation. If you are not understanding, I will explain once again. Okay? You no need to say please. I will explain.